Body Ban, and I will start with the lecture 3 about the signaling. What are the functions of the GPCR? GPCR function primarily by sensing molecules outside the cell and activating signal transduction pathways inside the cells. Here's an overview of their main functions. So the first one, I will talk about the signal transduction. GPCRs respond to the extracellular ligands like hormones, neurotransmitters, odorants by activation the intracellular G protein. So the ligands bind to the extracellular part of the G protein, and that is lead to the activation of the intracellular G protein. Open ligand binding GPCR undergo a conformational change, enabling them to interact with and activate G protein by facilitating the exchange of GDP to the GTP in the G protein. And then the activated G protein initiate various intracellular signaling cascade, and that is lead to the specific cellular responses. And that what we know is a signal transduction. Then the other function is amplification of the signals. GPCR protein, or we can say G protein, plays a role in amplifying cellular signals. A signal or a single activated receptor can activate multiple G protein. You know what is mean exactly by the amplification? It's like make it huge or make it bigger, So the single activated receptor can activate multiple G protein, each of which can in turn activate multiple downstream signaling, like cascade, multiple events happened now. And this amplification allows cells to respond robustly, strongly to even small amounts of the extracellular signals. That's mean maybe it's like a small signals, but the G protein make it a more amplification to it, and that this will lead to the huge response of these signals. It's not about like signal transduction, sometimes it may be amplification of these signals. It also recoagulation of the cellular responses. GPCR are involved in fine-tuning cellular response to the stimuli through mechanism such as receptor desensitization or internalization or down-recoagulation, which prevent overstimulation. Sometimes maybe that is leading to the amplification of the signals, but sometimes leading to the regulation of the cellular response, that is prevent the overstimulation. And also the other functions is modulation of the second messengers. Many G protein regulate the production of second messengers like a cyclic AMP or inositol triphosphate and calcium ions, which further propagate the signal within the cell to affect the specific responses. Sometimes make regulation of these second messengers, and this each one it has a function like a cyclic AMP. Lead to the activation of protein kinase A and IP3 that increasing the intracellular calcium ions, so that is affect the response of the cell. About the physiological, so we can say in brief, it's signal transduction, amplification of signals, recoagulation of cellular response, and modulations of second messengers. About the physiological rules of the GPCRs. GPCRs are involved in a number of physiological processes across different systems in the body, and some of the key rules include: the first one is nervous system. G protein are critical in neurotransmission, where they mediate responses to the neurotransmitters like dopamine, serotonin, glutamate, and they are involved in regulating mood, memory, sensation, perception, making them target for treatment for mental health disorders and neurodegenerative diseases. So because they are important in neurotransmission, so we can use them in treatment of the mental health disorders. Cardiovascular system. G protein also control the heart rate, the blood pressure by mediating responses to the adrenaline and non-adrenaline, because now we know that it has also the beta receptors. And we said before that is a beta 1 is responsible for the heart rate. They play a role in a vasodilation beta 2, for example, and vasoconstriction by acting on the receptors on the blood vessels walls, and that is control the blood pressure. Endocrine system, G protein mediate hormonal responses and regulate the release of hormones like insulin, glycogen, thyroid hormones. They also participate in maintaining glucose and energy balance, affecting metabolism and energy homeostasis. About the immune response or immune system, G protein are involved in immune cell chemotaxis, the movement of immune cells towards the infection site, and inflammation in immune cell activation. They help direct immune responses to the site of infection or tissue damage, making them critical for immune defense. Sensory system, G protein are essential for senses such as vision, smell, taste. For example, rhodopsin, which is type 1 of the G protein, that is present in the retina and that is necessary for vision by detecting the light. Olfactory receptors that is present in the nose detects the smell, and the taste receptors on the tongue detects the flavor molecules. About the reproductive system, G protein regulate reproductive hormones such as luteinizing hormone and follicular stimulating hormone, which they are essential for reproductions of a health and development. Gonadotrophin releasing hormone receptors, and they play roles in fertility, ovarian function, and spermatogenesis. Digestive system, G protein helps regulate gastrointestinal motility, secretion of digestive system, appetite, histamine receptor, GLBI1 CCK receptor. They influence hunger and satiety through the receptors in the hypothalamus and the gastrointestinal tract. So just show us how the G protein affects the various system, and we can say it's various system of the body. It's almost the system of our bodies. The G protein controls it, and that is making us the chance to discover drugs that is acting through the G proteins. Can you explain the way how a protein is secreted? Here's how the process works. The first one is mRNA translation. Ribosome binds to the mRNA in the cytoplasm, and then the protein synthesis begins. The co-translational pathway. Now we talking about the endoplasmic reticulum targeted translation. So the co-translation pathway, it's the ribosomes attach the endoplasmic reticulum membrane, and the protein is synthesized directly into the endoplasmic reticulum through something is called the translocator channel. The protein is fully made in the cytoplasm. Then something is called the chaperones keeps it unfolded for transport into the endoplasmic reticulum. Inside the endoplasmic reticulum, chaperones assist in folding. So the proteins may undergo chemical modification like glycolizations and something, and then segments of the protein embedded in the endoplasmic reticulum membrane and multiple sections form a mature transmembrane protein. So the first step is mRNA translation, and that is can be happen by ribosomes bind to the mRNA in the cytoplasm, and then the protein synthesis begins. Endoplasmic reticulum targeted translation, co-translational pathway. The ribosome then attach to the endoplasmic reticulum membrane, and the membrane synthesized directly into the endoplasmic reticulum through the translocator membrane. And the post-translational pathway, protein is fully made in the cytoplasm. The chaperones keep it unfolded for transport into the endoplasmic reticulum. Protein folding and modification inside the endoplasmic reticulum, and the chaperones assist in folding and maybe undergo chemical modification. And then the transmembrane protein, a simply segment of the protein, may be embedded in the endoplasmic reticulum membrane, or multiple sections form a mature membrane protein. To other questions is what is the main mechanism by which the receptor tyrosine kinase activated. The main mechanism by which receptor tyrosine kinase are activated, the first one is ligand binding. A signal protein ligand binds to the extracellular domain of two inactive receptor tyrosine kinase, bringing them together. This ligand induces dimerization of the receptor tyrosine kinase is a key activation step. It's an important step. Dimerization, like coupling of two subunits of the receptor tyrosine kinase. The binding of the ligand causes the two receptor molecules to dimerize or pair up, and this physical proximity activate the intracellular tyrosine kinase domains to each receptor kinase, and then that is lead to the trans autophosphorylation. The kinase domains of the receptor tyrosine kinase phosphorylate each other. That's what we call them as a autophosphorylation on a specific tyrosine residues in a process called the trans autophosphorylation. And this phosphorylation further activates the kinase activity of each receptor, and then it's lead to the downstream signaling. The phosphorylated tyrosine serves as a docking site for the various intracellular signaling proteins, which they relay and amplify the signal inside the cell, leading to the diverse cellular responses. And this photos activation, that is explain how the activation of receptor tyrosine kinase. This is the first is a single molecule. This is inactive receptor kinase, and then this is the dimerization that I said. Then it's bind to the receptors, and that is leading to the activation. Then you can see the phosphated group. That what we can say is autophosphorylation, and after autophosphorylation, then the downstream coupling that this can be happen. The fifth question is, to which group of protein RAS belongs? The RAS receptors belongs to the group of a protein known as a small GT base. And what is known as small GT base? It's like convert the GTP to the GTP. These proteins functions are molecular switches that alternate between the inactive GTP bound state and active GTP bound state. Guanine nucleotide exchange factor promote exchange of the GTP for GTP activating RAS. GTPase activating proteins enhance the intrinsic GTPase activity of RAS, and that is lead to the hydrolysis of GTP to the GTP and returning the RAS to its inactive state. RAS proteins play critical roles in signal transduction pathways and important in cancer conditions, particularly those controlling cell proliferation, differentiation, and survival. And by this way, we end the third lecture. Thank you so much for listening. I hope you understand.